everyone. I'm very glad that I have an opportunity to introduce our research, how to detect adaptive convergence in immune genes. Convergent evolution is a fascinating evolutionary mechanism when some traits may pop up many times in evolutionary history in non-related species. When you look at this figure, you see that each circle represents gene pool of one population, and by this arrow you can see ascendant-descendant relationship. And under the convergence, which is shown here in red, you may see that those allelic lineages evolve independently without having a direct common ancestor. Of course, convergence is not only one mechanism which may lead to the origin of shared polymorphism, which could be highly beneficial. However, the identification of convergence is quite challenging because convergence very often involve only a tiny percentage of the genome, and it may be also context dependent. However, convergence is still worth to studying, because if we know convergence, we can identify some common selection pressure. We can identify some molecular traits that have very high adaptive values. And also, we can better understand pathogen resistance. It has also implication for the breeding and pesticide resistance and for many other cases. While on phenotypic level, convergence is well documented, and it includes many textbook examples, as you can see here, here, or here. Its identification of the molecular level is pretty challenging, as I said. And what is even more challenging is to distinguish adaptive convergence from some neutral convergence. And one of the rare example that approach to this is evolution of ATP1 gene in herbivorous species, which is a gene which encodes a resistance against poisonous cardinalides that are secreted by some plants. And when you look at the species that are in orange, you see that they accumulate some convergently evolving or parallelly evolving substitutions. In immune genes, convergence was hypothesized to be particularly common because, as Ariane said, those genes are under very strong parasite-mediated selection, and some of them are even with direct contact with pathogenic molecules. And probably the best documented is the convergent evolution in MAC gene complex, where some approaches were developed to identify convergence based on the comparison of a peptide binding region versus a non-peptide binding region. And if we compare those trees, in case of any incongruence, this is traditionally considered as an indication for convergence. And the same is also true if we compare non-synonymous side trees with synonymous side trees. More recently, convergence was also detected by using amino acid haplotype networks in TLRs or based on surface charge clustering. However, all these approaches are to some extent system specific and cannot be widely applicable to any protein coding data. And to overcome this, we develop a universal methodological pipeline for testing evolutionary convergence in protein coding genes. And then we apply this approach to test how common is convergence in toll-like receptors in nearly 30 tits species. So we focus on toll-like receptors on two bacterial sensing TLRs, TLR4 that recognize lipopolysaccharide from gram-negative bacteria, and TLR5 that recognize flagellum from flagellated bacteria. And after the recognition, the signalization cascade is activated, and it provokes the secretion of pro-inflammatory cytokines that subsequently drive the inflammation as one of the major effector reaction of vertebrate immune system. And as the model group for studying convergence, we have selected tit family, which is a family of from small to middle-sized birds with well-known phylogeny and biogeography. And for sequencing, we use the classical amplicon-based sequencing using Illumina platform. Then we targeted the binding region of these both TLRs, and we gathered data set of nearly 30 species across the tit phylogeny. Since we observed quite extensive variation in nucleotide data, we first wanted to assess how to identify any like truly adaptive variation. So for doing so, we employed some test of positive selection, and based on the consensus, we identified some positively selected sites that we subsequently filter out if they evolve physiochemically non-conservatively. Parallelly, we also model out a three-dimensional structure of TLRs and evaluated whether these positively selected sites are located in close proximity. Then we evaluate their surface accessibility, allowing the interaction with ligands, and also assess whether they evolve convergently using a maximum parsimony evolution method. And also, we evaluated whether the surface charge and hydrophobicity also evolve convergently as a molecular phenotypic traits. And last but not least, we also evaluated post-translational modification. And the ultimate goal was identify some candidates for further potential testing. So looking at the results, we identified quite extensive like variation in TLRs with many positively selected sites. Those are shown here either in blue if they are identified based on the consensus of one method, or based on the consensus of more methods, which is in orange. And sites that are identified in other studies being functionally important are in black. And what can you clearly see here is that a lot of positively selected sites are located in the close proximity to the binding site. And more importantly, some of these sites also evolve convergently and pass all these aforementioned criteria. So all of this position can be further candidates for functional testing. And since toll-like receptors are heavily glycosylated molecules, and it was shown that this glycosylation may impede binding of particular ligands, or on the other hand, increase the binding repertoires of TLRs, we also assess whether the conversion occur in this post-translational modification. And we evidenced several cases of glycosylation, where several cases of convergence when the conversion was documented as an independent gain or loss of some motifs, which is shown, for example, here by this dark blue arrow. And then we also calculated the surface charge distribution on these TLRs, which is shown here for TLR4, and asked whether this surface charge distribution evolve in convergent manner. And for doing so, we compared the surface charge dendrogram with phylogenetic species 3. And as you can see here, there are a lot of discrepancies, which is an indication for convergent evolution. And in next step, we ask whether we are able to find any particular amino acid substitution that would explain the major surface charge cluster, because as we are working with mainly closely related species, it was better to condense all terminal branches into some surface charge clusters. And interestingly, we found amino acid substitution at this position, which has ancestor glutamic acid with negative charge that was mutated in some species into positively charged lysine, like independently, like several times. And this convergent evolving sites determined the major surface charge clustering in TLR4, which is an indication that molecular convergence can be manifested on molecular phenotype of this TLR molecule. And since we observe quite extensive convergence in TLRs, we also attempt to explain this convergence by some ecological traits, because as TLR evolution is mostly shaped by microbial communities. However, these communities are not known for our species, so we use some ecological traits that we believe that they could work as a good proxy of microbial communities. Those are listed here, And we applied RDA analysis while controlling for the effect of phylogeny. However, none of these ecological traits were present in the final model. There was only the weak effect of phylogeny. So how can we interpret this? So probably the ecological traits that we use are quite proxy of microbial community that shape TLR evolution. And also our results are quite well consistent with so-called co-diversification hypothesis, which assumes that phylogenetically more related host species may share related parasite communities. Basically, bacterial evolution just mirror its host evolution. To put it all into a nutshell, in our study, we identified that convergence occurs in TLR4 and TLR5 in specific amino acid position, and also in electrostatic surface charge. We identify some potentially convergent evolving site that could be a good candidate for further testing. As Adrian said, this is very important. And we were not able to explain any observed convergence in surface charge by the selected ecological traits. However, our newly developed approach can be quite widely applicable for testing molecular convergence, which is adaptive in any protein coding data. I would like to thank all the colleagues who are enrolled in this project, especially to my supervisor, Michal Vinkler, and you for your attention. And if you would like to learn more about this project, there is a paper that was recently published, and I'm also happy to answer any question during discussion.
So thank